I apologize for the short video there. I had a little technical difficulty, but I seem to have figured it out. So let's talk about our endomembrane system. The largest part of the endomembrane system is the endoplasmic reticulum. So the endoplasmic reticulum is a whole bunch of folded or tube-like membranes. So you can see sort of on the top left of the endomembrane there, the blue tubes that almost look like coral sticking up. Those are the smooth ER. They lack ribosomes on the outside. And then we have the rough ER that's sort of folded. I feel like they look like pita pockets in this picture. These folded pockets of membrane with little yellow or orange dots on them. Those are the ribosomes. They are stuck into the rough endoplasmic reticulum, and they're called rough because the ribosomes give them the appearance of being rough. So the endoplasmic reticulum is really an extensive network. It holds half the total volume of the membranes of eukaryotic cells, so it's quite large. And it consists of a lot of tubes and sacs that are called cisternae. So a cistern is a jar for holding water, and so that's where the name comes from. These little pockets hold cellular products. It is continuous with a nuclear envelope. That's the purple there. This book uses purple for the nucleus. So usually if you see a part of the cell with purple, it's the nucleus. Those pores there allow for proteins and RNA to move out of the cell and proteins to move in and out of the, the nucleus, and then proteins to move in as needed, because there's a lot of work that goes on maintaining and replicating DNA that requires proteins. So proteins need to move in, RNA needs to move out. We do see there are different sides to this endoplasmic reticulum. The cis side, if you remember, oops, I'm sorry, that's a different part of the membrane structure. Hold on to that thought for cis. But let's talk a little bit about the differences in the smooth and the rough ER. So in the smooth ER, we have a lot of metabolic processes. So lipids are produced there, carbohydrates are broken down, not converted into energy, that's a different cell, but can be broken down or built up there. There's also synthesis of steroids, sex hormones, things like that. The endoplasmic reticulum will also produce phospholipids for repairing or replacing parts of the membrane. So a lot of biological molecule synthesis and metabolism is going on there. One important function of the smooth ER is detox. So especially in liver cells, they have a high concentration of smooth ER. And in the smooth ER, the cell will sort of process out alcohol and other drugs and things like that. So that's really important for normal cell function. And then we also see some storage in the smooth ER. Calcium is a big one that's stored in muscle cells. So calcium's needed for producing the action potential, which we don't really talk about in this class, but the action potential for movement of muscles. So depending on the cell, the smooth ER might do more of one of these things and less of another, but it has all of those capabilities. So the ER, as I said, is also a membrane factory. So it produces phospholipids for repairing and replacing membrane. The rough ER has a related but different purpose, and that is for protein production. So we said that the rough ER has ribosomes all around the outside, and in those ribosomes we are producing proteins. So if the ribosome is producing proteins attached to the rough ER. Those proteins are usually going to be used within the cell. If the ribosomes are free-floating in the cytoplasm, or I'm sorry, the cytosol, those proteins are usually going to be excreted from the cell. So they're going to be something that's used outside of the cell in the organism. Another major function of the rough ER is that it's a membrane factory. So the smooth ER produces those phospholipids. The rough ER can assemble them into membrane structures. But the other important thing about the rough ER is that it can bud off what we call vesicles. So if you look down here in the bottom, we see this transport vesicle. This is sort of pinched off from the ER, and that little vesicle is like a little moving truck or a transport vehicle that's going to take products from the ER and move them around in the cell. Most of those products are going to go to an organelle called the Golgi apparatus. In the Golgi apparatus, the endoplasmic reticulum products are going to be modified. They're going to be sort of sorted, stored if necessary, and sent out to other destinations. So I like to think of this as like a packing and shipping store. It will add tags to molecules, finish protein folding, other things like that to prep any molecules that are going to be used in the cell. So those little vesicles will pinch off of the Golgi, off of the ER. We've got one here. It'll pinch off of the ER, and then it will fuse with the Golgi apparatus. And when it fuses, it dumps its contents into the Golgi apparatus. So the Golgi apparatus has two sides. The cis side, remember that term from chapter 3, cis means same. So on the same side as the ER, that's where it's going to accept those vesicles and accept the cellular product. And then those little molecules, usually proteins, are going to sort of process through the Golgi apparatus through a series of pinching off and fusing with the other folds in the Golgi. Eventually, they will be pinched off into a vesicle on the trans side, or the side away from the endoplasmic reticulum. Your book uses the same analogy of shipping and receiving. So on the cis side, the Golgi apparatus receives the molecule. On the trans side, it packages and ships out that modified molecule. When you look at this on a microscope, it looks like sort of a stack of flattened discs. Remember, these are all membranes. They're all part of the endomembrane system. Excuse me. Our next organelle is called the lysosome. So I like to think of the lysosomes as sort of the trash trucks of the cell. They basically break down and dispose of things, waste products or foreign things from the cell. You can also think of this as sort of the stomach of the cell, like the digestive organ of the cell. So the lysosome is very acidic, like our stomach, and it has a couple of different functions. It can break down macromolecules, and so it can either break down food type molecules or it can break down waste type molecules. So on the right, we see the process of phagocytosis. So that is when the cell brings in food, some sort of food molecule, by a vacuole. So a vacuole is a storage compartment. They can be used for many different things. In this case, we see the cell absorbing a food particle. It's coming in, the cell membrane sort of forms around it and produces a little pocket, sort of. And then it fuses with the lysosome. So that lysosome contains enzymes that are going to break down the food particles. And then the cell can use those products, those broken down products in the cell. The other job of the lysosome is called autophagy. So auto means self and phagy, or phago in the previous example, means eating. So it is breaking down the worn out organelles or damaged organelles from its own cell. And it works the same way. In this case, a little vesicle forms around the organelles that need to be broken down, fuses with the lysosome digests it, and then those parts of the cell can usually be reused in the cell, built into new organelles. Last, we have peroxisomes. These are real small organelles. They are used to break down and dispose of toxic compounds. They do that by producing hydrogen peroxide. Next, we get to vacuoles. So vacuoles are generally more common in plant cells, but they are sometimes present in eukaryotic cells as well as sort of a storage compartment. So they are storage compartments that are selective in what they store. Our example here in the picture is called the central vacuole. The central vacuole holds water in a plant cell. So the most common vacuole that we're going to encounter in our class is the central vacuole. And the central vacuole is important in keeping the cell full and keeping it healthy. So the cell wants to hold water in there, and it sort of keeps the cell nice and plump. We can also have food vacuoles. We saw that in the previous example with the lysosomes. Those are little storage compartments for food. We saw vacuoles also as part of the endomembrane system containing calcium. And then we also have something called the contractile vacuole. So a contractile vacuole is a vacuole that can pump things in and out. Again, this is usually water, and this is really important in organisms that need to regulate the amount of water coming in and out of their cells. Moving out of the endomembrane system, we have a few more organelles that we need to talk about. One is the mitochondria. So the mitochondria are really important because those are the organelles in the cell that take carbohydrates, take biological molecules, and break them down to produce ATP. ATP is the energy molecule in the cell. And nearly all eukaryotic cells have mitochondria. They all need a way to break down the sugars in the cell and produce ATP. Mitochondria use oxygen to do this, and we will talk about this process a little bit later. So I'm not going to talk too much about the process of cellular respiration, the idea of breaking down sugars or fats to produce ATP. And we're actually going to talk about the structure just a little bit because we'll come back to this one as well. Mitochondria are important because they contain their own DNA and they reproduce separately from the cell. So they're sort of special organelles. Inside the mitochondria is a highly folded membrane called the cristae. The cristae is the site of cellular respiration. So the proteins involved in cellular respiration are actually embedded in that membrane. And just like we saw with cell volumes, the cristae are highly folded, and that increases their surface area. And this is really important for the function of the mitochondria because it allows for more cellular respiration to occur in that small space. Another important feature of the mitochondria is that it is a double membrane. So if you remember when we looked at the prokaryotic cells, we saw that it had a cell membrane, and then it actually also has a cell wall or an outer membrane structure, not necessarily a cell wall. We'll talk about why that's important in just a minute. We also have the chloroplasts. These are found in plant cells. Mitochondria are found in all eukaryotic cells, plant and animal. Chloroplasts are only found in plant cells and other cells that are doing photosynthesis. So the chloroplasts are really important because photosynthesis is the process that sort of drives all of the energy in our living organisms. So in the process of photosynthesis, we're converting solar energy into chemical energy in the form of sugars. Those sugars can then be broken down either in the plant cell, or those sugars can be ingested by other organisms when that animal eats the plant, and then the mitochondria and their cells will break down those sugars. The main component of the chloroplast is the pigment chlorophyll. This is the molecule that's actually absorbing the sunlight energy and kicking off that process of photosynthesis. The chlorophyll is found in molecules called thylakoids, and if you look down on the bottom, you can see those green folded membranes, the thylakoids. Remember, structure equals function, so we're going to talk about those folds again. We saw those folds in the mitochondria, and we saw those folds in cell when we talked about cell size. Again, those folds increase the surface area. So the more surface area you have, the more chlorophyll molecules and proteins embedded in those chloroplasts that are going to perform photosynthesis. Chloroplasts also contain their own DNA, just like the mitochondria, and they also have a double outer membrane, or an inner and an outer membrane. The reason that I point out that these two organelles have DNA and double membranes is because of something called the endosymbiont theory. So it is suggested that the mitochondria and the chloroplast were originally bacteria or archaea-like organisms that were ingested by a cell, and they formed this sort of symbiotic relationship. So this slide here outlines the endosymbiont theory. You're welcome to pause it here if you want to take notes on it. I'm going to talk about it though with a figure on the next slide. So the endosymbiont theory says that an ancestor of a eukaryotic cell or a host cell that already had some organelles, already had a nucleus, already had an endoplasmic reticulum, engulfed a ancestor to the mitochondria. So this would have been a little bacterium or archaea that could use oxygen to produce ATP energy. And when that happened, instead of ingesting it, breaking it down, those two cells formed a symbiotic relationship. So a symbiotic relationship means a relationship that benefits both of the organisms. So the ancestor to the eukaryotic cell would have engulfed an ancestor to the mitochondrion and eventually evolved into this cell where the mitochondria is now dependent and part of the original eukaryotic cell, but still maintains its own DNA, its own double membrane, and performs a very specific function. So some of those cells went on to become the animal and fungi, and that actually should say unicellular eukaryotes, so went on to establish a eukaryotic line of organisms. At least one of those cells went off and decided to engulf something that was an ancestor to a chloroplast. So this would have been a photosynthetic bacteria. And just like in the mitochondria, instead of breaking it down, it formed a symbiotic relationship. So this would have gone on to evolve into the plant line, and the line that contains other photosynthetic cells. So all of the photosynthetic eukaryotic cells. Those cells have both the mitochondria and the chloroplast. So we know that the mitochondria would have been engulfed first because all eukaryotic cells have it, whereas only some eukaryotic cells have the chloroplasts. Again, the fact that these both contain their own DNA in the double membrane suggests that their ancestors at one time were fully functional separate organisms. So we've talked about all of the organelles inside the cell. Let's talk about the support system for the cell, the structure on the outside of the cell. Not on the outside, sort of on the outside of the inside. So the cytoskeleton is a network of fibers that supports and gives structure to the cell. So it runs through the cytoplasm. It's not on the outside. It's on the inside of the plasma membrane and also sort of pokes through the plasma membrane. But it can be sort of thought of as a scaffolding that is being used to support the cell. In that picture on the bottom, we have a cell. This is the nucleus. The yellow, or actually it's a bit more orange, the orange is cytoskeleton. The green is also cytoskeleton. These are two different types of cytoskeleton. And you can see that it's giving it sort of a volume and a structure. The main component of the cytoskeleton are the microtubules. So there are several other components of the cytoskeleton, but the microtubules are the part that really give it the structure. Just like scaffolding, the cytoskeleton can be deconstructed and moved. So one of the ways that cells are able to move is that the cytoskeleton can be broken down and move to another part of the cell, which changes the shape. Cytoskeleton is also important not only for the overall structure of the cell, but also for giving structure and support to the organelles in the cell. They can organize the cell, the organelles, they can anchor them in place, and they are also used for movement within the cell. So we have microtubules are sort of the main part of the cytoskeleton. We also have microfilaments and intermediate filaments. The microtubules are the largest components of the cytoskeleton, and they're sort of hollow rods. Another important function of the cytoskeleton is mobility or motility. The cytoskeleton serves as a track for moving things around inside the cell, but it can also serve as a mechanism of moving the cell as a whole. So there are two ways in which the cells can move. One is using cilia. So cilia are like small hairs around the outside of the cell, and they sort of beat together. If you think about rowers rowing a boat together, they all beat together and they move in a really smooth movement together. And then you have flagella. Flagella are usually found 1 or 2 per cell, whereas cilia are many all over the surface. The best example of flagella is on a sperm cell. When you think about the function of that flagella, it's to get the sperm to the egg. And the flagella are often 1 or 2, and they really have sort of erratic movement. Within the cell, specialized proteins called motor proteins will move things like vesicles around within the cell. So on the right side, we have a microtubule. We have a special motor protein that sort of clamps on to that microtubule, and on the other side is the vesicle. It requires energy, but that protein basically walks along that microtubule and moves the vesicle throughout the cytoplasm. So that sort of gives a directed movement for the vesicle. It can move from, say, the Golgi apparatus to the cell membrane or to another organelle in a directed way. Microtubules are also really important in chromosome movement. Chromosomes are packed down and condensed in the nucleus, and then during the process of cell division, they are separated in a very meticulous way and moved into excuse me, what will become the new nucleus, or the divided nucleus, and then what will become 2 new cells. Continuing to move out, we have the cell wall. So the cytoskeleton provides support internally, and then we have the plasma membrane around that, and then we will talk about the plasma membrane in the next chapter. Outside of that, in plants and also in bacteria, but we're talking about eukaryotic cells, we have the cell wall. The cell wall is really an external support structure. In plants, it's what gives the plant the ability to stand up against gravity. Animals have bones, plants have cell walls. It gives it structural support. It also helps to maintain the structure of the individual cells. So we've got structure for the cells and structure for the organism as a whole. The cell wall is made up of strong microfibrils. Those are basically ropes of cellulose. So remember, we talked about cellulose in chapter 3. Cellulose is repeating units of glucose that form hydrogen bonds with other chains of glucose. And remember, that makes for a really strong molecule. That cellulose is sort of woven almost into a mat, and so you get this really strong cell wall structure. Within the cell wall are openings called plasmodesmata. These are openings that allow for plant cells to communicate with one another. So no cell is isolated in a living organism, and so all cells have to be able to communicate between one another. We are not going into the section on cell-to-cell communication. It is quite interesting, but due to time constraints and just the level of what we talk about in this class, we won't be talking about how cells communicate. But it is important to know that they do communicate. So finally, we get to the outside of the animal cells. So in this picture here, we have a really good example of the plasma membrane. So that phospholipid bilayer around our cell. Inside, we have the cytoplasm with the microfilaments from the cytoplasm. So we've got some of that internal support structure. And then outside of the cell, we also have the extracellular matrix. So this is generally collagen fiber surrounding the cells, and it gives some outside support. So eukaryotic cells are basically made up of fats on the outside, on the plasma membrane, and so they need some sort of structure to give them a shape and support within the organism. So that extracellular matrix is connected to specialized proteins within the plasma membrane, which is how it sort of all becomes interconnected and has some strength and rigidity. The extracellular matrix is fully on the outside, so it's the outside support system compared to the the cytoskeleton on the inside. So that completes our lecture for chapter 4. This is the end of unit 1, so this is where we will end for our exam. And please make sure that you are doing your study guides. Let me know if you have any questions as you prep for your exam.